Start recording. And we're going to get started. All right, it says it's recording now, so I'm going to trust it. Oh boy, this one is going to be a long one. I've actually cut back on viruses for next class because most likely this is going to move into next class. So we're going to get started. I'm going to try to explain it all in the simplest terms possible for us all to understand. But this stuff is very crucial to moving forward in the class. We have to understand how bacteria grow. We have to understand a lot of their stress response mechanisms. And it's a lot of fun. Uh-oh, it's using timings. Let me get rid of this. Oh, come on. Okay, well, right now with everything else going, I'm not going to be able to do it. So we're going to start with chapter 4, bacterial growth and development. Here's all of the learning objectives. It's not the whole chapter. So a lot of the stuff in there about media and how we learn to count bacteria in the lab, it's stuff you needed to read. That's why I didn't take it out, because it's for lab, but we're not going to be covering it in lecture because we cover it so much in lab. All right, here's all the key terms, a lot of them again, but let's get started. So going back to what do cells need, what are cells in general responsible for, or what are they made up of, all that stuff. We break down their nutrition requirements into essential nutrients, macronutrients, and micronutrients. So essential nutrients are those that they have to be supplied to the organism from the environment. They can't make them on their own. Some bacteria can make virtually anything they need on their own. Others need a lot supplied to them. It just depends on the species. So one of these nutrients are the macronutrients. Macro meaning large molecule. So this is really looking at our biomolecules, our carbs, our lipids, our fats, our carbs, our lipids, our proteins, and we don't really use nucleic acids for energy. They're a little bit too hard for us to break down, so we don't eat that part of any organism that we consume. We don't eat it for energy. Now, the major elements that make up these macromolecules are carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur. The phosphorus comes from lots of inorganic material, but also it's found in our nucleic acids. That's why we need it. The sulfur is in our proteins. That's why we need that. And the rest, all of them have carbon, oxygen, hydrogen at the very least. Nucleic acids and proteins have a lot of nitrogen. The other ones don't necessarily have a lot of nitrogen in them. We also need lots of cations, positively charged ions. These are basically like cofactors for our enzymes, help them work better. So those are required materials for every single cell. Now, we also have micronutrients. What did I do now? And those micronutrients are thus named because, whoa, there we go. Because we don't need as many of them. So we just need trace amounts of these in order to survive and to be healthy, functioning cells. These includes a lot of our metals. So copper, cobalt, manganese, zinc, iron, all that good stuff that we will talk about more throughout this whole thing. And then we have things like sodium and chlorine ions. These are going to be really important for physiological processes, but bacteria don't use sodium nearly as much as we do. It's kind of like an alternative adaptation that they have if hydrogen ion availability is scarce, or if they're struggling in an alkaline environment, then they'll use sodium. Okay, any questions about requirements there? All right, let's go. So how do microbes build their biomass? So what is biomass? You remember? What does it sound like? Basically, yeah, its weight, how much mass it has. So how does it build its physical body and structure that has mass? And so all life on Earth is based on carbon, and that is basically what we build the rest of our mass around. Carbon is the central molecule for life, and we just kind of add other things to it and make it kind of fancy carbon. We make, sugary carbons and enzyme carbons and things like that. So there's two ways that organisms can acquire that carbon that they need. They can be autotrophs or heterotrophs. Autotrophs use inorganic carbon as their source, something like carbon dioxide. And so they can take it out of the atmosphere and turn it into their organic sugars that they need. Heterotrophs, these are the eaters. They get it from preformed organic molecules. So autotrophs take it from an inorganic source. Heterotrophs take it from an organic source already. And basically, this creates the balance in the carbon cycle. If we were all heterotrophs, eventually we would consume all organic molecules and convert them into inorganic CO2, and then all that carbon would be stuck in the atmosphere with nowhere to go. Autotrophs, if we only had those, eventually they would use up all the CO2 and have very little organic molecules for them to be able to use or to create from that loss of CO2. So they're constantly feeding into each other. And this is a key thing that we're going to learn a lot through this course is without bacteria in particular, a lot of these whole Earth cycles would cease to exist because they are the foundation for doing key chemical reactions that other organisms can't do. So this is how we get our carbon. The other thing that every organism needs to survive is have energy. How do we acquire energy? And this is where it gets a little bit more flavorful. So we can get energy from organic molecules. Those are organotrophs. That's us. We eat organic molecules. We eat food and we break them down. We steal the carbon from them because we're heterotrophs, but we also steal the electrons and the energy from those organic molecules. So organotrophs, us, this is the starting point. Then we have phototrophs. This is like your plants, your cyanobacteria. They may get their carbon from CO2 in the atmosphere, but where do they get their energy from? Well, that's light, photosynthesis. So we take the carbon and we have to break it down and then use the energy from light to put it all together to make sugars so we can build our body and so we can eat it. Then there are chemotrophs. So these ones get their energy from oxidation reduction reactions, redox reactions. You don't really have to know this too in depth, what a redox reaction is. So you don't have to remember how to do all the chemistry behind it, but you do have to know what the terms oxidation and reduction are, what's a reduced or a molecule, what's a oxidized molecule, that kind of stuff. Then we have lithotrophs. These ones use inorganic molecules as a source of electrons. So they're basically like rock eaters. That's what litho means, is rock, and troph means like feeding. So lithotrophs are going to eat the rock to get their electrons to power all their functions in their cell. So this is just how they do energy acquisition. We have to pair that with how they get carbon. So there's a bunch of different ways that we can get our carbon and our energy needs met. Any questions about this so far? All right, I'm going to move on then. So let's get into how we classify organisms based on both of these, their carbon and their energy acquisition. So the autotrophs are broken down into two types. We can be photoautotrophs or chemolithoautotrophs. So photo means light. So a photoautotroph gets their carbon from the atmosphere, CO2, and they use light for their energy source. That's like plants, cyanobacteria. Chemolithoautotrophs, we just call them lithotrophs just to make it a little bit easier for ourselves. They use that energy from the redox reactions to fix carbon from the atmosphere. So they're like plants in that they use CO2 and other inorganic carbon sources, but instead of using light energy to make the sugars, they use the electrons from redox reactions. So this is like our nitrifying bacteria that we're going to talk about in a moment. Basically, they steal the electrons from those redox reactions and do carbon fixation and all that fun stuff. Then we have the heterotrophs. The chemoheterotrophs, we call them just organotrophs to make it a little bit easier. This is us. So we eat organic molecules and we gain energy by oxidizing them, and then we use the carbon in them to build our biomass. We got to eat everything to get everything. And then there's photoheterotrophs. So this is a heterotroph, something that eats other things but gets energy from light. So here we're going to oxidize organic molecules to build our biomass, break down those organic molecules to get the carbon, but we don't break those down to also get energy. We get our energy from the sun. Just kind of weird, different things in the world out there. Now, the example here, purple non-sulfur bacteria, doesn't really help you, but there's very few of these in the world. So there's not many examples that we're familiar with that do this aside from those. I tried to give you examples for each of them so you can kind of put it in terms that you might understand. So the top two are very familiar to us and plants, and the bottom two are the ones that you kind of have to put into perspective of think about where do they get their carbon, where do they get their energy? Those are the two questions to figure these out. Any questions about that? All righty. Okay, so what this is basically showing is how this fits into the carbon cycle and metabolism. Right here you have central metabolism with glycolysis and the TCA cycle. And then over here we have our autotrophs with carbon fixation through like photosynthesis or something like that. And this is just going to show that it's all feeding into each other. It's a cycle. Without one or the other, the whole thing will break down. That's the whole point that you need to understand from that. I feel like we kind of gotten that since we were kids. These big geocycles are all interconnected. All right. Now, let's talk about energy again. We're going to kind of cross back to what we talked about before with membranes, but we're adding on to it now. So energy, as we said, can be stored in gradients across the membrane for later use. One of the main ones is a hydrogen ion gradient, or a proton gradient, because you take hydrogen, you remove its little electron, you're left with just a proton, and it's positively charged. So this creates not just a gradient of protons, but it also creates an electric gradient. So now we have way more positive charges on one side than the other. The cell, pretty much all cells, really likes to keep the inside of the cell very, very negatively charged, because this proton motive force, this gradient of hydrogen, is so important to us getting energy. If we had a very positive internal cell, then hydrogen ions wouldn't want to move in because they're positive. They don't want to add to the positive charge. So we try to keep the inside of the cell negatively charged so we can always use these protons for energy, and we call it proton motive force. There are tons of things in the cell that use proton motive force. Things like the efflux pumps that I studied, a lot of them will actually move the chemicals, the antibiotics, into the enzyme and then spit them out using ATP. Or I'm sorry, using proton motive force, not ATP. We use it to create ATP in the ATP synthase enzyme. We use it to move the flagella in bacteria. In us, it's a little bit different. We're more focusing on the bacteria side of things here. We use it for nutrient transport. We use proton motive force for so many things. So it's probably one of the main energy drivers in the cell. I know we hear ATP, and that is the energy molecule of the cell, but the other one is protons, and where we put them matters. So you might be a little confused about this too. How can just making a gradient equal energy? Think about it like a dam. If we push a lot of the water behind a big wall, and then the water slowly is allowed to leak out, we can capture the movement energy, that kinetic energy of the moving water, and use it to like spin a turbine or do some other form of work. That's the same idea. Think of it as not only are these molecules moving down the gradient, so they have kinetic energy that we can capture, but they're also charged, and the movement of charged particles can generate electricity. So we're getting electrical energy and kinetic energy out of this whole thing. Make sense? All right. So let's talk about the nitrogen cycle. Now we're stepping away from the carbon cycle a bit. Nitrogen is basically 80% of our atmosphere. And nitrogen, as we talked about last time, in the atmosphere is in the form of diatomic nitrogen, which has 3 covalent bonds between the 2 nitrogen atoms. That is very, very hard to break. Most living things cannot break that bond. The nitrogen fixers can. This is a class of bacteria that can convert that molecular nitrogen into things like ammonium ions, which all of a sudden now we can use because we just have single bonds here, not triple bonds. So we can break those a lot easier. Then you have nitrifiers, which can take that ammonia and turn it into something like nitrate, which is just another usable form that may be more accessible to other organisms than ammonia is. And then nothing is complete unless we can complete the cycle here. We also have denitrifiers, which can convert nitrates back into atmospheric nitrogen. So we can constantly move that nitrogen out of the atmosphere into the soil, back into the atmosphere. But without these nitrogen fixing bacteria, we would not be able to live. We need nitrogen to form our proteins and our DNA. And the only way we can get it is because of these nitrogen fixing bacteria. Otherwise, what we wait for some random lightning strike to break those bonds and put it into the soil by chance, that's not enough. So without bacteria, the whole nitrogen cycle would not be. No other organisms can do this stage here. So any questions about nitrogen fixation? We're going to talk all about this in the lab. It probably makes for one of our most complicated labs to understand what's happening, but it's probably one of the cool labs too, though, because of that. So how do we take up these nutrients that we've been talking about? Our membranes are designed to keep things out, but we need to eat. So how do we do that? Well, we get this selective permeability by 3 different methods, really. We can have permeases. These are selective carrier proteins. So they can say, I'm going to let glucose in, but not lactose. You got to go to a different carrier protein for that. You can have nutrient binding proteins. They're going to be in that periplasm between the 2 membranes of gram-negative bacteria. And they just kind of like scan the area looking for nutrients. Then they grab them and bring them to a type of transporter that can bring them into the cell. And then we have porins. These are going to be important to the outer membrane of gram-negatives, because without these porins, things often can't get through that outer membrane. So these are a little less specific just because we have another barrier before you get to the inside of the cell. So these channel proteins or porins tend to let a lot of not really specific things in, and then these nutrient binding proteins and the permeases will be more selective from that point on. So we want to get the stuff close to the cell, and then the number 1 and 2 decide what we really let in. Does that make sense? We're going to talk about these a little bit more, and we'll talk about some really cool ways that the cell brings things in. Sometimes I just learn this stuff and I'm like, wow, life is so cool. How did we do this? So the first one we've talked about briefly is facilitated diffusion. Diffusion is passive transport. It's just moving from a high concentration to a low, so no energy is required. But we talked about how polar and charged things can't get through the lipid bilayer. They don't want to pass through that non-polar hydrophobic space. So we just use channel proteins that don't use energy and allow things through. So how does that work? How can we get these things to allow something in without the use of energy? It's actually kind of cool. Right here is a glycerol transporter. The glycerol will be in a higher concentration in one area of the cell than the other. And as a result, this transporter will open up to the side that has the higher concentration. As the glycerol comes close, it will bind to the transporter just naturally because the shapes match, just kind of like an enzyme does. And when that happens completely naturally, just because the two molecules are now bound to each other, a conformation shift happens. Conformation is shape. So it goes from an open to the outside shape to now an open to the inside shape. And over time, just because of natural molecular movement, that glycerol will then detach and enter the inside of the cell. Now, because it's open on the inside as well, sometimes things can attach on the inside and move out as well. So we can have these work in both directions. If suddenly we have an excess of a molecule inside that we want to leave, these will work just as well. So we can bring it in when we want or let it out when we want. And no energy is being passed around at all. This is all just random movement following the laws of physics. And the whole shape change is just because they're binding to the substrate. And it will naturally change its shape as a result. And so this is just a top-down image of that. Or actually, this is an aquaporin, a little bit different than this one. And it also allows glycerol through. Any questions about facilitated diffusion? Facilitated just means help. So it's diffusion, but it needs a little help to cross the membrane. That's the way I think of it. All right. This is just basically everything I just said in word form. So I do that often because I want you guys to have it written out as well. I like to be able to point to the picture when I'm talking about it, but I feel like you should have this as well as the recording. Okay. Now, active transport does require energy, but energy is very, very sacred. And like all life, it's lazy. We want to conserve as much energy as possible. Prior to our modern day life, every single living thing is constantly worried about, where am I next getting energy? And life is just one food instance to the next food instance. So we have a lot of different strategies in our cells to minimize the use of energy wherever possible. One of these is coupled transport systems. So this is where we use these built up gradients and use that energy from the dam to move things across the cell. And we do this in two different ways. The first way is symport. Sym means together. So this is transporting things together in the same direction. So what we see here is we have a high concentration of red solutes outside the cell and a low concentration inside, and it's the opposite for the blue. So if I wanted to move the blue against their concentration gradient, that would require energy. I don't want to use ATP because that's a very valuable energy source. So because there's already a gradient of the red guys here, I can capture that movement flowing energy of them down their gradient to kind of sneak one of the blue ones in too. So we couple the transport of something against its gradient with the transport of something with its gradient, and they're both going in the same direction in this case. So the two molecules will travel in the same direction, into the cell or out of the cell, both at the same way. And we just use these transporters that don't require the use of ATP or any other type of energy source other than the flow of the red down its gradient. Then there's antiport. Anti means against, so this is transporting against the direction. So here we're still moving one thing against its concentration and the other with its concentration gradient, but now they're going in opposite directions. So now the red has a higher amount inside the cell than out, so it's going to be transported out of the cell down its concentration gradient. We're going to capture that flowing movement energy to move the blue, which is lower outside, into the cell. So just the two molecules move in the opposite directions, but the idea is still the same. You've learned this in cell bio already, right? Okay. The problem is we all learn these things before. It's a matter of remembering. So if I'm going a little bit back to basics, it's because the basics are that important. And if you don't get them, someone needs to explain them to you. And sometimes you understand one professor's way of explaining better than another. So I feel like I got to review that stuff with you. Okay. But sometimes that's not enough. We do need ATP to do things. That's why we have it. So in this case, we can use ATP transport through ABC transporters. Now that's not just a fun, oh, ABC, how cute. It stands for ATP binding cassette superfamily. These are cool. These are little enzymes that embed in the membrane, and they're found in all domains of life. We have them, plants have them, bacteria have them, archaea have them, everybody has them. And what they do is they take a material up against its concentration gradient by using ATP. So the bottom part here, this yellow part, is the part that binds the ATP, and through that binding, it allows the solute to enter and be transported into or out of the cell. So we can take them and use them to take up nutrients. We can also take them and use them to expel waste or other things like antibiotics that we don't want. So one that I've studied is right here, MAC-AB Tol-C. And instead of just being one single membrane protein and having a porin that lets things into the periplasm, this one just connects all of it. The outer transport porin of the Tol-C allows connection to the ATP binding cassette. And so things actually are in here, antibiotics that we don't want. They bind to the ABC transporter, and ATP is used to push it into the outer porin, and then it just leaves the cell. So it's basically like saying this stuff is really, really bad. We don't want it to get caught in the periplasm, so we're going to force you to completely leave the cell. But otherwise, we just have these unselective porins. Materials can come in, and then these soluble binding proteins can bind to the solute and bring it to the ABC transporter. So there's a lot of different ways we can do this. These are not quite as common as the ones that only span one membrane. These are actually a lot more rare. Any questions about this? Okay, let's keep going. This is just basically the same idea of what I just said. Sorry, I do that a lot. I apologize. Does that help you guys, or would you rather me not put those extra word slides in at all? Okay, and I don't come to these sometimes too because the picture will be bigger on these slides, and here you'll be too busy reading it to listen to what I'm saying. Okay, next is siderophores. These are cool. Iron is so important to life. It's a very important cofactor, but most iron in nature is not in a form that life can use. And so we have to capture the very, very, very little bit of iron that is in a usable form. We don't really have a nice, tricky nitrogen fixing type bacteria that can convert it into a usable form so that it's plentiful. In this case, iron is and always will be a very limiting nutrient. So we're all fighting for it all the time. bacteria have these things called siderophores. And these are specialized molecules that bind to that rare form of iron in nature. They bind in a way that we call chelation. So if you ever hear that term, chelation just means to bind, and it binds in a very specific way. It forms these what we call coordinate bonds. And basically a coordinate bond is a really weird covalent bond. Instead of sharing the electrons, one of them just says, no, 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 take them all, take them all. And now my nucleus is positive, so just you have extra negative charges, bind to my positive nucleus. So it's actually a really, really strong bond. I'm not going to make you explain that, but you're going to hear these terms, coordinate bond, chelation, and I just want you to know what they mean. It's a very, very strong covalent-like bond. And what this will do, they will exude or expel these siderophores. They'll release them into the environment. It will form these really strong bonds to the iron, and then it will import them back into the cell. It can import into the periplasmic space and then get further imported into the cell, or if this is a gram-positive, it just gets imported into the cytoplasm. And when it's released into the cytoplasm, it has to be converted from an Fe+3 to an Fe+2 form that's a little bit more usable for us. And we have different types of enzymes that can do that. So sometimes we have multiple different siderophores that can move it across all these different forms or spaces. Sometimes we just have one that ferries it all the way in. It just depends on the cell. Now, some bacteria like to get their iron from us rather than competing for it in the soil. Things like the mycoplasmas or Neisseria gonorrhoeae, they actually don't have siderophores often. If they're a cell that likes to live inside our cells, they don't want to release siderophores because that's just one more thing for our immune system to attack. So instead, they'll put receptors on the surface of their cell that kind of like, as the cell's moving around, they're like, ooh, there's some iron that's in this cell. I'm going to grab it and I'm going to steal it from the cell. So that's just one other method that you can use to get this. And one of the reasons why we have fevers when we are sick, not just because one, the increase in temperature makes our enzymes work better to kill them, everything moves a little bit faster to the area that it needs to get to, but fever causes our iron to take up any free metals that are floating in our body. So zinc, manganese, iron, all these really important enzyme cofactors, fever will bring them in, store them in the liver, and make it really hard for these cells to get it. So then they starve themselves for these key nutrients and they die. That's the idea, at least. So lots of reasons why we have fever. This is just one you've probably never heard of before. Any questions about siderophores? All right. This is cool. And I know reading it in the book at first, you're probably like, this is confusing. I promise you it's not. It's really, really cool. So group translocation is a really neat process that uses energy to chemically alter the substrate during its transport. All right, let's talk about what that means exactly. We're going to use one example of group location here, the phosphotransferase system. And this is found in all bacteria. And what they do is they use a phosphate group from PEP, phosphoenol pyruvate. This is a intermediate of glycolysis, so we're making it all the time. And it has lots of phosphate groups on it that we can kind of snag and use for other things. So they take a phosphate group and stick it on specific sugars to change their chemical structure. What this basically says is, I want to bring glucose into the cell. But eventually, if I bring in too much glucose, now I have changed up my gradient, where there was more outside and less inside. If I take it in a lot, now I have more glucose inside my cell than out, and it's going to try to leave my cell. I don't want that to happen because nutrients are super, super scarce. I really want to make sure that the glucose stays in me until I'm ready to use it to make energy out of it. So maybe I don't make it glucose anymore. Instead of bringing glucose in and letting it sit in my cell, as it comes in, I steal one of these phosphate groups, stick it on the glucose, and guess what? You're not glucose anymore, you're glucose 6-phosphate. So now you don't have a gradient anymore for that, because glucose will constantly want to come in, because every time it comes in, it stops being glucose. So to the gradient, there's no glucose at all in the cell. Glucose then always wants to rush into the cell. It's kind of a really sneaky way of making sure that you never have an opposite gradient for your food sources. So we do this with our sugars a lot, because sugars are what we turn into ATP. And so we have a modular design that allows it to work on all different types of sugar transporters. So what does this look like? Here we have an example of a glucose transporter, and here's a mannitol transporter, just two different types of simple sugars. And as the glucose or the mannitol comes in, we have this whole system of enzymes here that takes a phosphate from PEP and transfers it to the incoming glucose. So as it's being transported in, it gets turned into glucose 6-phosphate. So not even for a second is glucose inside the cell. Does that make sense? This whole enzyme thing can just also attach to the mannitol transporter and do the same thing for mannitol. It can transfer to a lactose transporter and do the same exact thing to lactose. It's basically assuring we never have a situation where there's more sugar inside the cell than out. And then we don't have to rush to break this down. We can wait a minute. Does that make sense? Now, how is this energy neutral? Because what we're doing here is called phosphate level, or substrate level phosphorylation, excuse me. Words are hard today. That is effectively an energy neutral process. You don't have to understand that. That's more cell bio stuff. But just know that substrate level phosphorylation, the moving of a phosphate group to another item, another molecule, is basically an energy neutral process in this situation. So this whole thing doesn't take up any energy to effectively move things against their gradient. Does that make sense now? Does someone need me to explain it a different way? It's okay if you do, because this is important. This will be on the test. Probably a short answer question. I'm trying to like really hammer it home, like, here's your chance, ask me questions. Okay. So that's just explaining the same thing. Now, here's a quick question for you from the textbook. So which of the following statements regarding this phosphotransferase system, the PTS, is or are true? A, the system has a modular design that accommodates different substrates. B, the substrate being transported is altered during passage into the cell. C, it's a form of active transport driven by ATP. D, A and B, E, A, B, C. So take a minute, talk to a friend about it if you need to. What is true? Which statement is, or which statements are true? We'll break it down together. Okay. So A says the system has a modular design that accommodates different substrates, different sugars. Is that true? Yes. Yeah, we can use it for glucose, we can use it for mannitol, we can use it for lactose, all those different things. True. The substrate being transported is altered during passage into the cell. Is that true? Yeah, we're adding that phosphate to it while it's being transferred. It's a form of active transport driven by ATP. No, that's the whole point of this one. We don't want to use our ATP in this case. We're lazy. All cells are lazy all the time. So they're basically saying, I'm going to use this substrate level phosphorylation to pop over a phosphate group, and no other energy is required to do this. Otherwise, everything is just going to be gradients moving things into the cell. So we're moving glucose from higher outside to lower inside, always, because there's never glucose in the cell because I keep adding phosphate groups to it and that turns it into something else. So in this case, D is the correct answer. A and B are correct. C is not because we're not using ATP directly to do any of this. Or indirectly in that case, but. All right, we good? Cool. So we're gonna switch topics a little bit here. And I'm gonna kind of go through a portion of this a little quickly because we're hitting it hard in lab and we're not gonna focus on it too much in here. It's more a lab concept. And that's the bacterial growth cycle. So as we talked about, bacteria divide by binary fission. They replicate their chromosome, then they form a septum, and then the cells separate. Sometimes that is one cell separating into two. Sometimes bacteria do this really cool thing where the cell will just keep getting bigger and bigger and bigger, and they're doing like 10 different replications inside, and then all of a sudden, bam, you have 10 cells. But overall, the way that the bacteria grow is through exponential growth. They double. One cell becomes two cells become four become eight. We're always doubling in number because of how they divide. Now, some can divide asymmetrically. So we have this weird hyphomicrobium that divides by what we call budding. We don't really need to know those weird examples or exceptions in this. That's the only type your book brings up. So I just thought I'd bring it up to just say there are other ways that cells divide, but we're just focusing on this binary fission to produce exponential growth. So exponential growth looks like this first graph A. As we start, we have 1 that becomes 2, 2 that becomes 4, and suddenly we're doing these gigantic numbers. And we're effectively constantly increasing the rate at which we grow because the numbers keep getting bigger and bigger so fast. If we want to do any math with these types of graphs, we convert them into logarithmic graphs because then this converts this curve into a straight line and we can do easier math to find the slope, which is the growth rate. So in class, this is the one we're going to care about a little bit more. And when we look at the bacterial growth in general, it forms this really interesting curve that you see in C here. And it's divided into 4 phases. Lag phase, right here, the line is flat, meaning our cell number is staying the same. Why do you think we'd have a lag phase? Why is the cell number staying the same at first? This would be if we're like moving it to a new location or putting the cells into new medium, something like that. Why do you think they stay the same? Yeah, they have to acclimate. They have to, stop and be like, okay, how is this environment different than my last environment? And in this lag phase, they change up their gene expression. They say, okay, I don't need these genes to be turned on anymore. Let's stop doing those and turn on these ones instead. Maybe you go from an environment where lactose is the sugar that you're bringing in, and now you're suddenly in a glucose environment, and glucose is always preferred. So we got to switch over to all of our glucose mechanisms. Then once we've done that, once we're ready to go, we start dividing right away, and the cells immediately jump into log phase. This is also called exponential phase. It's also called growth phase. It's the phase where they are actively doubling, where the 1 becomes 2, 2 becomes 4, blah, blah, blah. And then they enter into a stationary phase. So cells can die here. They can enter a state of dormancy, and they can still, kind of divide. They just don't do it very often. For the most part, stationary phase, they're no longer growing. They're just maintaining. And this is because they've gotten signals that say the environment doesn't have quite as much food as before, so let's slow down our growth rate or we're going to starve ourselves here. And this is basically where all bacteria live. If they're inside us, if they're in the environment, they're basically wanting to stay in stationary phase whenever possible. So a lot of really cool things happen in stationary phase or late log phase that don't happen in any other stage of the growth cycle. And then we have death phase. This will also be exponential, and this is where the bacteria have completely depleted all nutrients. They've cannibalized as many of their neighbors as they can for parts. They are building up toxins in their environment from their waste, and they can't survive the waste toxins anymore, and they can't help it. They're going to start dying. This can be a months-long process because bacteria can be very dormant. They don't need to be constantly growing or doing any type of gene expression, so they can slow that down to really survive these harsh times in case the environment suddenly becomes great again, because bacteria are constantly having changing environments. So those are the 4 phases. Any questions about those? Okay. This is all you really need to know about it. Everything else we're going to talk about in lab class. So don't stress too much about these different growth phases. So let's take a pause. Let's keep talking about some cool bacteria. I know you guys are like, they're not that cool, but I think they are. I think they're cute. I think they're fun. Alrighty. Biofilms. These are really cool specialized structures. Basically, they're communities of bacteria. Normally, we think of bacteria as these like lone wolves that just go out and do their own thing, but they can actually come together and form communities and sometimes even begin to behave like a multicellular organism, all in these biofilms. And they will form biofilms on pretty much anything in nature, including us, because we are also nature. So if you've ever been in a creek and you slip on the rocks, rocks aren't supposed to be slippery. They're slippery because there's like biofilm growing on them. All of us have had plaque on our teeth at some point or another. Plaque is a biofilm of bacteria growing on our teeth. Eating all the sugars that we take in, it's a nice little place for them. So these can be made of just one species. A biofilm can be all a singular species of organism, or it can be cross domains, and many species can join in this biofilm together. It's very fascinating stuff. Now, at the end of log phase, and right as they're about to get into stationary phase, bacteria will start something called quorum sensing. And this allows them to do things like form biofilms and perform group coordinated activities. What happens with quorum sensing, biosensing? Bacteria are constantly secreting chemicals that are going to correspond to an action that the cell can take. But because they're constantly secreting them, they don't want to bind their own signal and start doing something when they're not supposed to. So since they're always secreting it, these chemicals won't bind and begin to cause a change in the genetic expression until the signal reaches a certain concentration. Usually that corresponds to a specific size of the bacterial population. So once the bacteria hits a certain population size, they're producing enough of these chemicals passively that they can start binding to these surface molecules and create a response. One of these responses is a really cool example here. Shoot, I cannot remember the name of this squid, bobbinose squid. They have a little pouch inside of them where they let bacteria grow. And every morning, they reach in with one of their tentacles and scoop out most of the bacteria and get rid of it. And then they go about their day. But then, right when the sun is about to go down, the bacteria have been growing all day and they reach a population where they've hit that quorum. And they start to turn on bioluminescence. So they start to glow, only on the bottom of the squid, not on the top. Why? That way the squid can come to the surface of the water and eat, and it'll just look like the glowing moon to anything underneath it. But during the day, it doesn't want to be shiny and attract attention, so it has to go in and scoop out the bacteria, remove the quorum, and now the population has dropped so low, it can't glow. This is kind of a really cool 2 organisms that are completely different working in concert together. It seems smart, I don't know. But that's one example of biofilms. Another one is, I thought I put it on here. They happen a lot. Quorum sensing happens a lot in our bodies as well. When bacteria reach a certain level during an infection, they will suddenly say, okay, we've established ourselves. Let's turn on all the horrible things that make this person sick. We call those virulence factors. They'll release toxins. They'll start attaching to things and forming their biofilms and just creating lots of problems for us. They don't want to do that too early because a lot of these virulence factors will set off our immune system. They want to wait till their numbers are high enough that they could potentially overwhelm our immune system. So that's another cool use of quorum sensing. So why form biofilms? Well, sometimes it's better to be together. It's better to be protected and in a bigger group. And when we really think about it, yes, these bacteria are all individuals, but they're all mostly clones of each other. So bacteria do kind of want to look out for the bacteria next to them if it's one of their clones. So they will act a little altruistically and help these other cells because basically it's meaning that their genes also survive. So, for example, when nutrients are plentiful, bacteria will adhere to a surface and start to form a biofilm because basically they're saying, hey, this spot's pretty good. It's full of food. I don't want to leave. Then when the nutrients deplete, they start to break away and dissolve the biofilm to go look for a new place with food. It could be food, it could be other things. Sometimes they form biofilms to respond to a stressor. So when there's a lot of antibiotics present in our body, they may start forming biofilms because the thick, gloopy covering that they put over themselves of polysaccharides prevents antibiotics from getting in. So it could be, hey, this is a great place to be, or we got to hunker down and protect ourselves for a while. There's many different ways that that they will form biofilms. Let's talk about how the biofilm forms, though. There's 5 different steps. First, it starts with what we call planktonic bacteria, like plankton. They're just free swimming. That's what that means. And then one of them will be like, hey, this is a nice place to form a biofilm, or, oh no, there's a stressor, I need to form a biofilm. And they will start to stick to a surface. They can do this by using the pilus. I know we didn't talk about that one. It was in the book, though, from last lecture. It's basically a projection on the surface that allows them to stick to things. So they can do that and start to stick. Then what they will also do is start to secrete a polysaccharide. Sugars in general are very, very sticky. So they secrete this polysaccharide and it sticks them to the place, and it also allows other bacteria to come in and stick better as well. Then as they start to grow in number, they will start to create this exopolysaccharide, EPS. And it's a really thick polysaccharide layer that starts to form all the way around them and protect them. It once again protects from antibiotics. It can make sure that they all have the ability to exchange nutrients with each other, things like that. And then the biofilm starts to form these really interesting shapes. Often it'll be really skinny at the bottom and then kind of create like a balloon shape on the top. And that allows water and nutrients to pass through even on the bottom, because inside biofilms, if you are very, very deep in the biofilm, you're not going to be getting nutrients as good as ones close to where the nutrients are. It'll take time for them to diffuse to you. And they have strategies to cope with that too. Then, if for some reason the biofilm is no longer useful or protective, or they're running out of nutrients, some will start to secrete enzymes that break away the EPS, that break away the exopolysaccharide, and then they start to swim away again. Now, they can also attach with their flagella. They can do this kind of like twitching motility that kind of like moves along the surface until they find a good place to attach. There's a lot of cool, interesting things beyond that. That's all in chapter 12. I'm not going to make you know that stuff. It's just kind of cool to know. If you want to learn about that stuff more, I suggest you take microbial physiology. He gets into that stuff. All right, so as I said, the inside of a biofilm is going to have a very different behavior than the outside, where we have nutrients or oxygen. Sometimes we don't want to be exposed to oxygen and we want to be on the inside. So bacteria will have a differentiation. What we mean by that is the cells behave differently based on where they are in the biofilm. Just like our cells, they all started out as a zygote, and over time they differentiated into a liver cell, a bone cell, all these different things for different purposes. Same idea in here. So this is a picture of an E. coli colony. This down here is where they'd be touching the media with all the nutrients. And so the nutrients come in the bottom here, and E. coli don't need oxygen to grow. So in here, they're growing very nicely. But up here, they're kind of starved for nutrients because the nutrients have to travel all the way up that stalk in order to get to these ones. So these guys are actually in stationary phase, while these guys will be more in exponential phase. Now, some bacteria, what they will do is they'll kind of do like a timeshare. They'll say, okay, we have the nutrients for now, and then in 30 minutes, we're going to start transporting the nutrients up to you, and you can grow for a while and we'll stop growing, and then we're going to take the nutrients again. So they have these very interesting, very complex ways of making sure the whole community stays alive. And sometimes they just say, okay, well, we're not going to grow. You can grow, and you're just going to protect us. So there's lots of cool things that they do. There are other forms of cell differentiation. Sorry, I was trying to think of how I'm going to talk about this stuff. Bacteria are constantly facing environmental stressors, and so they have to learn to adapt to them in many different ways. Biofilms are one of them. The one way that we're going to talk about in depth now are endospores. The rest of these I'm not going to talk about, but please read about them in the books, especially the heterocysts of cyanobacteria. That's a really big one. I'd rather us get to other things today, and the book explains these pretty well. So read those, make sure you know them for the exam. But let's talk about the endospore, because this is probably the more important one. Gram-positives, some gram-positives, not all, can do this really cool thing where they form an endospore. This is basically like taking their cell and putting it in a capsule and saying, we're going to go into this dormant phase. We're basically like cryo-freezing ourselves to shoot into space for a while. That's the idea here. They're going to form a spore, hide all their genetic content in there, and let it live in a really harsh environment, protected from the environment until things turn good again, and then it will germinate out again. So the whole process is called endosporulation. And we call it an endospore because it's being formed inside the cell. The whole process takes about 8 hours, and it initiates in starvation conditions, not just like I'm a little hungry. This is like I've been hunkered down in my house or in Costco for 2 years now. I've eaten the whole neighborhood. I'm the last human alive kind of incidents. We're starving to death here. This is last chance. And what they will do is they will start to divide asymmetrically and then save one of the divisions as the spore and then sacrifice the other half. And we'll get into that. It's mainly done by two genre of bacteria, Clostridium and Bacillus. Clostridium, this is the important one for humans. This is Clostridium tetani, does tetanus. Clostridium botulinum, botulism. They cause C. diff. It can cause lots of like ulcerative colitis. It can cause gangrene and wounds. These spores cause lots of problems in the health world, and we'll talk about that more hopefully at the end of the semester. If not, take Med Micro. We talk about them a lot there. All right, let's talk about how we start creating a spore. Stage 0, you have your vegetative cell. This is a normally dividing cell. It's just another term for it. And it will decide, this is anthropomorphizing it, they don't really have higher brain functions like us, but they get signals saying extreme starvation environment, and this gets triggered. So it chooses one side of the cell to begin septation. In stage 1, the chromosome is going to replicate like normal binary fission, except for one thing. Normally, when we copy our chromosome, the origin just splits but stays on one side. In this case, the origins are going to migrate to completely opposite sides of the cell. And this means while it's being copied, you're going to have this piece of DNA crossing the entire cell, and we call that the axial filament. And you can see it under the microscope while it's happening. If you look at like a, not a light microscope, a electron microscope. And so we started this replication process, and then stage 2 begins. This is where the septum starts to divide, but it's not going to be in the middle. It's going to be somewhat unequal. We're going to form one really small compartment and one larger compartment. And this will start to occur before we've completed replication. And replication is mostly happening on one side, the bigger side of the septum. So right before that septum finishes closing up and splitting off the cell has to quickly take DNA polymerase and like shove the new chromosome into the little hole that's quickly closing up. Why they do it that way, I don't know. So we make a 4 spore with the new chromosome and the smaller portion, and the rest of it is known as the mother cell, the one that's creating the endospore. Then we have divided into these 2 separate cells. The cell wall is still intact. We've just kind of divided that inner membrane, or they don't have an outer membrane. So we've just divided the membrane at this point. Then we go into stage 3. In this case, the membrane of the mother cell will actually start to push out and engulf the 4 spore. So it kind of like creates a second membrane around the 4 spore. Kind of like what we did when we took in the bacteria to become our mitochondria and the chloroplasts. And this is all inside the peptidoglycan cell wall. In stage 6, the mother cell's chromosome begins to disintegrate. While this is happening, the mother cell is directing, make tons of peptidoglycan, make tons of protective proteins, and surround that little 4 spore. So it's trying to create all these things to protect the 4 spore while it degrades its chromosome. Let me keep going. Then in stage 6, the cell will make a lot of dipicolonic acid. What this does is inside the 4 spore, it's going to reduce the water content from 70% down to 30 or sometimes 10% water. So this is one of the key ways that a spore can survive, because water is pretty corrosive. It's constantly causing chemical reactions. So if we decrease the amount of water available, our molecules are going to stay more intact. They're not going to be degraded by water. We're not going to have to constantly be fixing them and putting them together. We also are going to stabilize and protect the spore's DNA with this dipicolinic acid. Then calcium is taken up by the coat of the spore, this big, thick cell wall-like structure that's being formed. The spore coat makes this 4 spore resistant to heat, resistant to drying out. It can even go into space and come back and be like, yeah, that was no big deal. Where there's lots of radiation and there's a vacuum, it makes this thing super resistant. And then in stage 7, the mother cell begins to break apart. It has completely done everything it needs to coat and protect this little spore. And so the mother cell breaks apart and releases the spore. And now that spore can live wherever it is. Live is a very loose term. It can survive wherever it is for 50, 100 years, potentially longer. And basically nothing can touch it until the conditions are good again, and then it can germinate out and become a dividing vegetative cell again. So that's the whole process. Any questions about that? It's pretty cool. It's like an ultimate saving mechanism. I am going to start the next one, the next chapter, because we were supposed to do 2 here. Chapter 5 is environmental influences on growth. We definitely will not finish this one, but we will have enough time next class to finish it. Once again, we're not going through every single thing in the chapter, but just a lot of it. Lots of key terms in this one, but only 1 page, not 2. All right. So we have to start this off with a discussion of what's normal. What are normal growth conditions? We have designated normal to be sea level pressure, temperatures from about 20 degrees Celsius, which is like in the high 60s, to 40 degrees Celsius, which is like high 90s. Then a near neutral pH, so we want to be close to 7, as close as we can, 0.9% salt content, and lots of nutrients. This is not really the norm. This is just what we've designated the norm. In reality, a lot of these organisms that we're talking about, bacteria, never evolved to live in these conditions. These conditions developed relatively late in Earth's geological time frame. Really early Earth was a very extreme world. So a lot of these things that we're talking about as being stress adaptations were just the way that these cells lived for years, and then they had to adapt and evolve to handle normal conditions. But we are a very self-centered culture and people, so we say this is normal, everything else is the weird one. And it just allows us to see, okay, if this is how we survive, what are some ways that things survive outside of how we survive? Because we really are doing science in a lot of ways to learn more about ourselves. So anything outside of that range is an extremophile. They live in extreme environments. A lot of them are archaea. We do have bacteria that are extremophiles, some eukaryotes. The vast majority are the archaea. So let's talk about them. We have lots of ways that we designate them based on their temperature, pH, preferences, all that stuff. We'll go through some of these now. The oxygen one, the designations are not super important for this class. That's lab, but how they survive different oxygen situations will be important for this class. So let's start with temperature. So bacterial cell temperature matches its environment. It's kind of like cold-blooded, ectothermic, not like us where we control our cell temperature all the time. So as a result, if it gets super cold, their membrane starts to harden up, they can't get nutrients in or waste out as easily, and they gotta adapt to that. If it gets super hot, their membrane starts getting super melty, and too many things can get in and out. So they have to survive in situations that otherwise would harm the cell. And every organism has an optimum growth range. There's one temperature where they're like, this is the sweet spot, everything's working at optimum. I can handle a little hotter, I can handle a little colder, so there's my range of maximum and minimum, but outside of that, my cell starts to die really, really fast. So they grow fastest at the temperature where their proteins work most efficiently. That's the story of surviving extremes. Are your proteins happy? If your proteins and your membrane aren't happy, your cell's not going to be able to grow and it's going to die. So that's where we're focusing on a lot here. And growth will stop when the temperature causes critical failure to the membrane or the proteins. So let's talk about the different ways that we categorize these guys. Psychrophiles, not psycho, psychrophiles, grow from about 0 to 20 degrees Celsius. So about right above freezing temperatures. Psychrotrophs, you might hear this term too. These are organisms that are really mesophiles. They prefer to grow between 15 and 45 C. This is like our body temperature and normal ambient temperature. So they prefer to grow there, but they can handle the cold temperatures pretty well. They just don't like growing at them. So they grow best at mesophile temperatures, but can live just fine there. That's something like listeria that can infect cold meat products, processed meat products, and it can grow in your fridge. That's why it's dangerous, because it's like, yeah, it's not ideal, but I can still grow here, and then your food spoils and gets you sick. And some Yersinia species that can cause plague, they can also do this. Then we have thermophiles. They like it hot. So 40 degrees C is, we are 37.5. So this is like right around high 90s, 100. And 80 degrees C, that's very, very close to boiling. That's 20 degrees away from boiling. So these guys are living in hot temperatures. Hyperthermophiles, they can live at extremely high temperatures, well above boiling. And they usually do this because they're also under high pressure. So it keeps the water from boiling off into steam, and it keeps it as liquid water. So when we're talking about these two, already we can see if you're living in one extreme environment, most likely you're also living in a second type of extreme environment, and you have to have the ability to tolerate both. So we get these growth curves where we see there's an optimum growth rate for each of these, but also a range. The ranges can overlap, but their optimums are pretty unique. Any questions about that? Mesophiles, if you notice, their optimum is basically our body temperature. These are the guys that like to infect us. Those are the ones that make us sick. Yeah. No, you don't have to know the range, just know generalities. So like cold, freezing temperatures, body temperature, little hotter than body temperature, very, very, very hot boiling water. Yeah. All right, so yeah, I am going to go over this with you. You do not have to understand this graph. This is something you will learn in biochemistry, and it's talking about enzyme kinetics and how well the enzyme is functioning, and they've tied it back to the growth rate of the organism. So what we see down here at the bottom is the enzyme functioning in blue. The black is the optimum growth rate of this bacteria. So at lower temperatures, the growth rate or the functionality of your enzyme is not at your growth rate. So you're not growing well. This is partially because at low temperatures, your membrane is stiffer, so you can't get as many nutrients in. And you also have fewer enzyme substrate collisions. Everything temperature-based means, are my molecules vibrating fast or are they vibrating slow? The slower things vibrate, the less they're going to move around, the fewer collisions there's going to be. And remember, nothing in nature, except for our thinking, is purposeful. Organisms are all because random chemicals bounce into each other and happen to react because they hit at just the right way, at just the right speed. So if you're not hitting as much, you're not going to grow as much because your enzymes aren't doing anything. Then we start to hit this sweet spot where the enzymes are functioning well enough to match our optimum growth rate. And as we keep getting a little bit hotter, we keep growing a little bit faster and faster because our enzymes are moving faster. And then we hit a point where we start to diverge from that optimum again, and we start to grow slower. And it kind of seems like we're doing okay at first, and then it just falls off a cliff. This is at the higher temperatures where, yes, everything's moving really fast, things are getting in and out great, but now my enzymes can't survive. They're starting to denature because of the heat. When that happens, they don't have the proper shape, they can't do their proper chemical reaction anymore, so we can't live. And they can kind of tolerate that for a little while. They have mechanisms in place to keep themselves stable, but after a certain point, either time spent at that high temperature or the temperature goes even higher, all those things start to fail. So we have ways of survival a little bit outside our window, but not a lot outside our window. That's what that's saying. So how do we survive high temperatures? Every cell has what's called a heat shock response. So once again, we can study heat shock response in bacteria and know what to look for in our own cells. Then we can look in our cells and say, yep, it does the same thing. And now we know how we can tolerate physiological stresses of temperature better and how we can, help ourselves become better people, basically. So we learn a lot from bacteria. The heat shock response is a global response. This is a term that's often used in bacterial genetics. Global means we are affecting many different sets of genes, not just like these are heat response genes. No, we're going to talk about membrane genes. We're going to talk about DNA replication and transcription and translation genes. We're going to talk about tons of other genes being affected now. So it's a global response. And why does it occur? Because we notice that our proteins start to misfold. When they're misfolding, they're not doing their job anymore. We're not getting something done, so we say, hey, we gotta send this out to fix it. And what they do is they produce a lot of chaperones. Chaperones are special proteins, special enzymes that go out and target these misfolded proteins and say, let me just put you right back and kind of fold you back into the right position. And okay, you can survive a little bit longer. And then find another misfolded protein and put it back in its correct shape. Or it can move things where they need to move in the cell. Chaperones do a lot of cool things. They also create enzymes that change up our lipid composition, like what we talked about way at the beginning. We can alter all of our membrane lipids to stiffen or loosen up our membrane and make it more fluid based on what we need. Yeah. That triggers the heat shock response, which then triggers a bunch of different genes to turn on or off. And that includes things like creating chaperone proteins and enzymes that help change up our whole membrane. It can also cause us to slow down transcription, translation, all that stuff, so that way we don't keep making broken proteins, things like that. And once again, this occurs in all living organisms. Any questions about heat shock? I'm going to go through pressure really quick just because it's simple. So sea level pressure is 1 atm, 0.101 megapascals, or 14 pounds per square inch. You don't need to know that. Just we're using it as a way to measure, okay, where are these other guys surviving at? Barophiles, another term for them is piezophiles or piezophiles, I've heard it both ways. They can grow up to 1,000 atmospheres. 101 megapascals, 14,000 psi. That's a lot of pressure. It's very hard to break those kinds of cells open. A lot of times we need to break them open to get to their parts to study them. These guys aren't going to break no matter what we do to them. They can survive pretty heavy pressure. We have to break them a different way, not just pressure-wise. And then we have barotolerant organisms, which can grow from about 1 to 50 megapascals. So a little bit more to kind of a lot more, but not quite 101 megapascals. And they can grow okay in that, well in that phase, but then as it starts getting more and more pressure, their growth fails pretty quickly after that. All right. A lot of times you'll see these barophiles or barotolerant organisms at the bottom of the ocean near the deep sea vents where it's also very, very, very hot. So you'll see hyperthermophiles and barophiles in the same area. Often they're the same organism.